It is my pleasure to introduce you Dr. Matthew Hill. Matthew Hill is an associate professor at the Department of Psychiatry at the University of Calgary. He received his PhD at the University of British Columbia in Vancouver with Dr. Boris Burzalka. Then he moved to New York with Dr. Bruce McEwen to study stress and endocannabinoid. So I had the privilege to meet Matthew about 10 or 15 years ago when he was still a PhD student in British Columbia, and he was a very, very dynamic person. He completed his PhD with about 25, 30 papers. At the end of PhD, he was already highly cited. His work on endocannabinoid, stress, endocannabinoid, and mood has been very, very important, inspired a lot of research. In 2002-2003, when Matthew started researching endocannabinoid and mood, there was just 3-4 papers about this topic. Now we have 1,000 papers explaining the mechanism of endocannabinoid in mood regulation and stress. We are very lucky in Canada to have this very dynamic, very intelligent person, and now he's really important person in the field of endocannabinoid and mood disorder and stress. Matthew.
Well, thanks a lot, Gabriella, for that really kind introduction. As you can see, well, actually you can't because the title slide. What do I press?
Yes,
Oh, that? Okay. So as you can see, the title of what I'm going to talk about today is a little different than both of what was in the program, as well as what my area of research primarily is, which is really in the domain of stress. I've dabbled a little bit looking at endocannabinoids during development and in adolescence. So I think this will be kind of an interesting presentation. I'm going to give you the short punchline at the beginning, which is, unfortunately, I don't have an answer to this question, as I don't think anyone does, because the evidence is confusing and contradictory, and there's a lot of reasons why, although I do hope to point out a few things I think we can move forward to answer this question in a more meaningful manner. Okay, so as Gabriella had pointed out, we should disclose our potential conflicts. So like Robin, I also have had some interactions with GW for some consulting, and we've had some grants off of them. And I also have done some consulting with Pfizer. None of that has anything to do with what I'm talking about today, because that's all in the domain of anxiety stuff. Okay, just to review what the learning objectives would be here, I think the first thing I thought that might be good to do is to kind of review the ontogeny of the endocannabinoid system and just explain its development and how it changes through development in the brain, and some early signals that it might be important for certain aspects of brain development, because I think that kind of sets the stage for further discussion. I'm going to kind of in a very brief overview way just discuss some of the animal and human studies that have looked at cannabinoids on brain development when there's exposure during the adolescent period. But I'm going to, more importantly, I think, point out what I view as some of the major limitations in this area and things that need to be addressed for moving forward if we actually want to get the answers to the kind of questions that we're asking about here. And that's the third aim, is to really kind of highlight where I think the research needs to go. So it may not be in this three-part, it's kind of integrated all together, but those are the main points of what I'm going to go through today. So I think the first thing I'd like to start with is to just discuss what we kind of know about how the endocannabinoid system develops and what it does in the brain. So usually, especially being the third speaker in, I thought there would be like an endocannabinoid introduction, so I didn't actually do a lot to kind of throw you the biology of the endocannabinoid system. But in the easiest form, this is kind of the way that we understand as neuroscientists the way that the endocannabinoid system functions. It's a retrograde signaling system where endocannabinoid molecules are predominantly synthesized in the postsynaptic neuron, and they act as feedback signals that gate neurotransmitter release. And usually these molecules are produced in response to cellular depolarization, like in the state of, say, epilepsy or hyperexcitability. You get a big dump of endocannabinoids that try and feed back to the presynaptic terminal to turn off glutamate release. So it's generally a retrograde signaling system, and its main role is to tweak synaptic transmission and mediate a lot of forms of synaptic plasticity, like long-term depression, long-term potentiation, and stuff like that. But one of the things that we've also learned, this is kind of the canonical understanding of what endocannabinoids do, but we also know that during neurodevelopment, especially in early life in the fetal brain, where a lot of this work has been done, that endocannabinoids actually have a secondary purpose, which is they act as axon guidance signals. And so a lot of really beautiful work from Tibor Harkany has kind of laid out the fact that a lot of these axonal growth cones, as the axons from neurons during development go to form circuits, they release 2-AG as almost a chemoattractant signal, 2-AG being one of the primary endocannabinoids. And this helps guide axons to make sure that they go to their appropriate place and form the appropriate circuits. This is really important for cortical to subcortical circuits. They've done some beautiful work disrupting this signal and shown that you don't get appropriate corticothalamic inputs, for example, which has been the basis for some of the work that he's done with Yasmin Hurd to look at the possible effects of prenatal exposure to THC and how it might interfere with this process. But this is an important thing to understand because while this has been studied predominantly in early life, we are fully aware now that the brain does continue to develop well on beyond gestation, and it goes into adolescence now. And so some work from B.J. Casey, who I think is a phenomenal cognitive neuroscientist, she's done some really great science looking at adolescent brain development, and she was one of the first people that really pioneered this idea that brain development is not linear. There was a group of people working at NIH in kind of the late 90s that came up with these findings that showed that it wasn't necessarily a straight line that brain development occurred on. And in fact, this kind of underestimates some of what we actually know now about how areas like the prefrontal cortex develop during adolescence, because what we know is that there's actually an overproduction of synapses and then a subsequent pruning event. It's almost like the groundwork gets laid for a multitude of potentials, and based on life experience, that is then refined down to a more formative prefrontal cortex that matures for that individual as they go into adulthood. But this is usually by adolescence, what we know is most of the subcortical structures seem to have reached their mature state. But areas, especially like the prefrontal cortex, which is going to be important because that's a big focus of a lot of the stuff with cannabinoids, do seem to continue going through development through the adolescent years. And they used to say till about 18, now that's been even updated, and now say it's probably closer to 25 that the prefrontal cortex is developing until. So the question then that comes along with this is, since we know endocannabinoids are very important in circuit formation and neurodevelopment in the fetal period, is there actually a role for endocannabinoids for prefrontal cortical maturation and development through the adolescent period? So this is just a quick slide to put everyone on the same terms, because there's a lot of I'd say confusion and debate to some degree about developmental stages between, say, a rodent, which is, I'm a basic neuroscientist, so I do a lot of work with rodents, and humans, because obviously the lifespans are quite different. But this is a general picture of the different lifespans that we look at and how they typically are defined. I would say this would agree with maybe 85% of people who study development in animals. I'm looking for a nod from Dominique. I don't know. But basically what you see is when a rodent is born, it actually has, its stage of neurodevelopment is more similar to about a second trimester going to third trimester human baby. And they tend to be equivalent around P11 is around the point where you're at the same level of neuronal maturation that you'd see with a human. But then you have these kind of in a rodent's very short truncated periods like childhood or a juvenile period into adolescence. I'd say the majority of the field kind of looks at adolescence as this peripubertal time. They look at when the animal would, for example, leave the nest and move on from weaning, which is usually around the late 20s, around P30 in age. So for a rodent, we kind of roughly define the window of P30 to P60 as an adolescent window, at least to capture up until about age 18. And that's roughly based on neurodevelopment, the kind of window that we're talking about. So that window of P30 to P60 is very important if we're trying to understand adolescent brain maturation. And so we're going to focus mostly on this window here. So this is just a graph from work that I had actually done during my postdoc years ago in Bruce McEwen's lab, but we still haven't published it, so that's never a good thing when you've been out of someone's lab for 7 years. It's written now, though. But basically, we were mapping the ontogeny of the endocannabinoid system, and we were looking at how that was influenced by exposure to early life stress. What I'm showing here is just the control data of what anandamide levels, being an endocannabinoid, look like in the prefrontal cortex. And what we see here is that at birth, in a very early neonatal stages, anandamide levels are actually really, really quite low. And what you see is what looks like, I wouldn't call it a linear. We are obviously missing days, and this is not scaled appropriately for the time. But it's a semi-linear increase where you tend to see anandamide levels increase from early life into adulthood. But what we kind of skipped over here, and this was obviously an error on our part because we weren't as attuned to the field, and this was consistent with actually a lot of what's been done in human postmortal tissue, is we missed this, we didn't really focus on this whole period of adolescence. So a lot of the human work that's looked at the ontogeny of the cannabinoid system has looked in fetal or neonatal tissue and then adults as well. So there's a lot of knowledge that we do not have about how the cannabinoid system in the human brain, for example, evolves through the adolescent period. So we're starting to learn a little bit more about the endocannabinoid system in rodents by focusing on this period. So to kind of zoom in on that window there, a fellow graduate student of mine, Tiffany Lee, that we were both working in the same lab, and she continued this project after I left grad school, but she was really looking at fluctuations throughout the adolescent period and how endocannabinoid levels were changing. So I'm just focusing on anandamide here because of the fact that it's the endocannabinoid that we tend to study more. Also, we didn't see dramatic changes in 2-AG, which is the other endocannabinoid levels. But also, as we move forward, I'll point out one of the reasons I'm focusing on this more. So on the left, we have anandamide content, and what you see is these kind of waves of anandamide that seem to occur during the peri-adolescent period. And this has been replicated and found also by Yasmin Hurd and her group down in New York, as well as an Italian group, Daniela Perolero and Tiziana Rubino, have found very similar fluctuations through various adolescent windows. And it's interesting because the spike in anandamide that happens around P35 is actually, there's been some suggestion it might relate to pubertal onset because it does tend to correlate in females quite nicely with sexual maturation. No one's definitively looked at this, although it has always been a lingering question that we've had over the relevance of endocannabinoids to shifting some of those hypothalamic circuits to kind of wake up and move into a pubescent state. But what we do know is these waves of endocannabinoids are very strange, and we're not exactly sure why they happen the way they do. Anandamide is metabolized by an enzyme called fatty acid amide hydrolase, or FA, which this graph over here is depicting the activity of this enzyme. And we see roughly an inverse change in patterns throughout adolescence that we see with anandamide. And what I mean by that is, as anandamide levels go up, the activity of its metabolic enzyme FA goes down. So we do believe that these changes in anandamide that are happening throughout the adolescent period, these waves that we see, are actually mediated by shifts in its hydrolysis by its enzyme FA. We also see very interesting changes in CB1 receptor expression throughout development. And so this was a study done by Kwai Tsang, who's at Rosalind Franklin. And what they did was they actually did a series of approaches looking at both in situ hybridization of the message RNA for the CB1 receptor, but they also did a series of electrophysiological studies which pretty much validated the exact same finding they have here, which is if you look in the prefrontal cortex on glutamatergic neurons, so obviously the excitatory input to it's coming to a lot of these cortical neurons, what you see is throughout development this progressive loss of CB1 receptors on the glutamatergic afferents that come into the prefrontal cortex. And this is interesting because this does suggest that there is going to be a differential effect of cannabinoid receptor activation on a developing brain based on the neuronal populations that express CB1 receptor versus a mature adult brain, where actually in the cortex, the predominance of CB1 is actually on GABA terminals and not on glutamate terminals. So this is an important point to think of, I think, because what it does suggest is there is these ontogenetic changes in the system which could be important for understanding what happens if THC hits the system at various developmental windows. But I think also a question that I'm more interested in as a basic scientist is, do these changes in endocannabinoid fluctuations that occur during the adolescent period contribute to any of the ongoing brain development that is actually happening in that same window? And so I wish there were a lot more evidence to actually provide you on answers on this, but I'm going to highlight two studies, one human and one animal, that I think are really interesting and really important in this regard. So the first one is a study from B.J. Casey's lab, and this came out in PNAS last year. And this was building off of a finding we'd had on a collaborative project a few years ago, looking at this gene SNP, or a genetic variant, in the FA enzyme. So FA, which is the enzyme that metabolizes anandamide, within human populations is a naturally occurring SNP in the gene for this enzyme. And if you're a carrier of the rare allele, which is an A, so there's a substitution of a C for an A, it actually results in enhanced proteolytic degradation of the enzyme, so that people who carry this enzyme actually have less FA and higher levels of anandamide. So given that we don't actually have any tools in humans yet that we can study dynamic changes in anandamide signaling, say giving a FA inhibitor, they are in clinical trials, but there's nothing available for experimental purposes. There's not a lot we can always definitively say about what anandamide's doing in humans, which is one of the reasons why this gene SNP and FA has been very interesting for us to study. So a few years ago, we had collaborated with BJ and her colleague Francis Lee because they were developing a mouse version of this FA SNP. And one of the things that they found in both the humans and the animals was that there was enhanced functional and structural connectivity between the prefrontal cortex and the amygdala that seemed to relate to individuals having the rare allele, so having more anandamide, exhibiting lower levels of anxiety and enhanced levels of fear extinction. So their thought was that having this enzyme actually resulted in a gain of function with more cortical top-down control of the prefrontal cortex over the amygdala. So they then followed this up, both of them being interested in developmental processes, and actually looked at this phenotype across age. And so with I think there's about 1,100 individuals in this study that they collapsed together, ranging from really young kids of about 3 up to about 20 years of age. And what they found was if you look at the maturation of the white matter tracts coming out of the prefrontal cortex to the amygdala, you have more, a greater density of the white matter tracts from the prefrontal cortex to the amygdala in individuals that bear the FA SNP that gives them higher levels of anandamide. And so you can see the aggregated data on the left. This is the histograms of the FA for the analysis, but you can see the spread on the right. And what's really interesting about this is this appears to be an adolescent phenotype that does not emerge prior to about the age of 12. And you can see the inflection point on the scatter plot looks like it's happening around pubertal onset. They did the same study in animals and found the exact same data that was in P25, kind of a pre-weaning or a pre-adolescent at least animal. You saw no differences in the white matter tracts or the connectivity. And as you go through the adolescent period, as this projection pathway develops, you start to see greater density in those that have this allele. So the suggestion from this is actually that yes, levels of anandamide do play an important role in the outgrowth of axons coming from the prefrontal cortex to subcortical areas like the amygdala, and it facilitates the neurodevelopment of this process. So I think that's actually some really interesting evidence. The other side of the coin is from Daniele Perilero's lab in Italy, where they did a series of studies where they decided to block the CB1 receptor using a CB1 receptor antagonist during discrete windows of adolescence. And what I'm showing here is just a single figure when they did a block period between the ages of P46 to 60. They did it at 3 separate windows. They did P25 or P30 to 45, 46 to 60, and then 60 to 75. But no matter how they manipulated at each point, they found the same kind of shift in the way that the glutamatergic system in the prefrontal cortex was maturing. So this is that process I was speaking of where you have pruning, where you have an overproduction of synapses, and then you have a pruning of those synapses back. And what they were basically showing, looking at protein levels of glutamate receptors, but they saw the same thing with postsynaptic density measures, which are, in theory, a marker of synaptic density. Was the same thing, was you actually abrogated this pruning process. And the waves of pruning that would normally happen during the adolescent period were modulated if you took the CB1 receptor offline. So that does again suggest that the endocannabinoid system is playing a role in the developmental processes that are occurring in this still maturing prefrontal cortex. Okay, so just to summarize the little bit I said there, what we know definitively is that endocannabinoid signaling does modulate axonal growth and circuit formation, especially cortical to subcortical circuits, in addition to its kind of established role as a modulator of synaptic transmission. We know that endocannabinoid signaling does exhibit dynamic fluctuations through adolescence. The functional role of this we have not quite figured out. No one's really established this yet. In fact, there's actually very few people studying this. And we know that from the study that came out of B.J. Casey's lab that augmentation of anandamide CB1 signaling seems to at least relate to enhanced white matter connectivity between the prefrontal cortex and the amygdala in humans. And we know from the work from Daniele Perilero's lab that disruption of endocannabinoid signaling during discrete adolescent windows can modulate the maturation of prefrontal glutamatergic synapses. So that does kind of at least set the stage that we know that there is an ontogeny of this system during a discrete developmental period, and that it seems to be playing a role in various aspects of neurodevelopment, particularly with respect to the prefrontal cortex. Okay, so then we move into the preclinical studies where we actually look at the consequences of cannabinoid administration during discrete windows of adolescence in animals. So I'm not going to belabor this because, to be honest, there's dozens of these studies. And the problem with aggregating the data out of them is that everyone uses different drugs, they use different doses, different routes of administration, and different time windows. So it's not easy to give you a general take-home message that's from this. I will say the primary findings that people seem to report are adverse generally. These are often countered by a lot of findings that are negative and a few findings that actually show positive outcomes. But they generally suggest that there's alterations in brain development. So this has been found. Therese J, as well as Yasmin Hurd, as well as Daniela Peralero have all looked at spine density on prefrontal cortical neurons and all shown THC during adolescence seems to cause a reduction in dendritic spines, suggesting that there is some change there. And this is a picture from the paper that came out of the Peralero lab. So there is this suggestion that there's compromised brain development. And as Robin had alluded to in his talk, a lot of animal studies do seem to show these kind of residual impairments in cognition, as well as some behavioral indices of emotionality, like heightened anxiety or depression-like behaviors. Now, I'm going to kind of pull out one point before I go on to that. I do think it's important to point out, actually, so regardless of what I'm going to go into next, which I view as the methodological constraints on these studies, is the fact that I do think it's very interesting that, regardless of what I say next, that these effects do seem to be specific to the adolescent brain. And when you do these studies in humans or in adults, often these effects are not seen the same. So this would suggest that there is something specific about the developing brain. So it does suggest that there is some sensitivity there. But we have some, what I would view as very significant methodological issues that can compromise some of the translation of these data. And I mean, I'm throwing myself under the bus because some of these papers involve stuff that I've done myself, so this isn't, I'm openly recognizing the flaws, and I think this is the problem we see in science. It's an evolving field, and a lot of things we didn't know 10 years ago, we do now, that I think are very relevant for understanding these. So I'm going to call this a little bit lost in translation. And I think there are three main points that we need to consider. And I think for anyone who's doing cannabinoid work, especially on the developing brain, these are the questions we need to ask. Because it's one thing to tailor a study to ensure that you see an effect. And there's another thing to tailor a study to try and mimic reality if you're actually trying to generate data that's going to have translational relevance to understanding what's happening in humans. So I think the first issue that we've encountered, and this is one that I've done myself because I've used some of these drugs in animal studies, is that synthetic cannabinoids, and Robin alluded to some of these that have come out, like the John Huffman compounds, but also the ones that are more typically used, like WIN 5552212 or CP 55940 or HU210, we now recognize that these are actually very different in the way that these molecules activate the CB1 receptor than THC does. And I mean, Robin had pointed out THC is a partial agonist. These molecules are almost entirely full agonists. But outside of the partial versus full, what we now have learned as well, especially through some really nice work that's come out of Mel Kelly and Eileen Donovan-Wright's lab at Dalhousie, is that actually the intracellular signaling cascades that these molecules activate are very different from one another. And so this is just an example here where you can see, looking at changes in phosphorylation of the ERK cascade versus phospholipase C, or over here AKT, we see that the effects of THC are actually almost in the opposite direction from what we see using CP 55940, which is a drug that a lot of us have used before doing these studies. So what I mean by this is, while we have seen effects from using these drugs, and they may relate specifically to activation of CB1 receptors during developmental processes, whether those are relevant for any changes we would have seen in humans consuming cannabis, I now, at this point in my scientific career, would say is questionable. And I say that because I'm not clear that they're even evoking the same cellular effects that THC would. So I think that's an important point to recognize when we evaluate these studies. The second one is, as has already been discussed today multiple times, there's a lot more to cannabis than just THC. And as Robin had pointed out, we know, especially with something like CBD, there are a lot of other molecules that are in cannabis that, when it's consumed as a whole plant, are fundamentally different than THC in isolation. So, for example, I think it's very interesting, the studies that have been done in humans using IV THC, for example. If you do injectable THC in animals, it's almost always viewed as aversive. They find it an aversive experience. Versus if you actually do pulmonary exposure with a vapor, animals find it rewarding. So that in and of itself, I think, is a big point to begin with. But that's just a route of administration. I'm going to talk about that in the next slide. But there's also this big difference between looking at THC in isolation versus whole cannabis. Because as Robin pointed out, even the presence of CBD, there are several reports now that have shown moderating effects of the impact of THC by the presence of CBD. I would say in Canada, it's probably true that our street cannabis has no CBD in it pretty much at this point. I believe our cannabis strains are somewhere around 20% on the street, more so They seem to even be a little higher than what they see in London. But yeah, our current cannabis, anyone who's buying street cannabis, you're probably getting almost no CBD in that. But that being said, there are a whole host of other molecules that we actually don't understand their interaction. But the basis for a lot of people who use cannabis regularly and claim all these fundamental differences between strains, this has not really been scientifically evaluated, so I never take much of that with more than a grain of salt. But the reality is there are all these other molecules in there, the terpenes as well as about 70-odd cannabinoid molecules, whose biology we have not studied in any meaningful depth. And we do not understand how they interact. And I think, again, if we're going to be trying to model and understand how cannabis use for a teenager is going to be impacting the brain, and we want to use a preclinical model, then we need to be modeling that by using whole cannabis to actually replicate what we're looking at and not just an isolated component of it. I think there's value in looking at the effects of THC, certainly, but I think if we really want to be able to pull evidence out of this, we need to know the whole picture. And then the third point, which is what I already addressed, is this difference in route of administration. And so I just put these pictures up because this was a study that Paul Mallet did, who's over at Wilfrid Laurier, and he actually was the study I just referred to about looking at the condition-place aversion versus condition-place preference was the one who demonstrated if you use the volcano approach for vapors and you gave animal access to that, they found it rewarding and they developed a place preference to where they would get cannabis vapor. If you injected THC in the animal, it developed a profound aversion. And most people who've been trying to study cannabis dependence or cannabis abuse in animals have had a massive problem in this because most animals do not like to self-administer cannabis through intravenous routes. And the only way you can usually get to do it is by pre-exposing the animals to cannabinoids to some degree, possibly by downregulating the system a little bit. As we move forward, we're starting to understand that actually the smoking option or a vaping option does produce fundamentally different effects, as Paul found in his paper. And so my argument is if we want to actually develop preclinical evidence that we can use in the capacity to have information about what is happening to the developing brain, we need to model appropriately routes of administration, dosing, and the whole context of cannabis as opposed to the isolation of THC. So a colleague of mine, Ryan McLaughlin, who actually did his postdoc here, first with Gabriella and then with Dominique, has moved on and he has his own lab now down in Washington State. And these are photos that Ryan provided because he's one of the first people I've seen who's put a lot of effort into actually trying to develop an animal model of self-administration of cannabis. And this did not work with an IV approach, but what he found here, and these were actually professional photos they had done for a whole piece, he just got an NIH grant to do this, was using this approach where you have a vapor system, he can actually train rats to lever press to get hits of cannabis vapor. Now, I think that this is fascinating because this now opens the door to studying different aspects of factors that could influence cannabis use and abuse, like looking at if an animal has early life stress or concurrent stress, does that make them self-administer more? Looking at other variables, but from a perspective of looking at adolescent brain development, this is really good because Ryan's been able to show he can titer THC levels to exactly match those that are in humans from recreational cannabis use. You can scale it so you can look at low levels versus excess use. So you have across the board to model different aspects of it. But importantly, he's also modeling entire cannabis. They're doing cannabis extracts and oils, the same things that you can go and buy at the stores in Washington. Now this is granted a little easier for him because he does live in a state where cannabis is legal. So it has given him an advantage over a lot of his American colleagues now. But I really think this is the way forward if we want to get evidence that we can say definitively would translate in a meaningful capacity on the effects of cannabis on the developing brain. Okay, so that's the animal stuff. So onto the human stuff. And it's not like this is less fraught with problems than the animal stuff. All this research is complicated. I would say in human studies, the majority of the studies that have been done to date employ retrospective cross-sectional approaches where they talk to adults and they compare those that smoked cannabis as a teenager versus those that didn't. There's clearly a lot of value from these studies because it can be very meaningful to understand different outcomes, but at the same time, a lot of the data that's been generated from these cross-sectional studies has not replicated well across study to study. And this seems to be due to a lot of factors. The ones that seem to be the most predominant, though, that have come up again and again are pre-existing differences in cannabis users that were there before they started consuming cannabis. Differences in early life socioeconomic status. So if someone grew up in a low SES versus if someone grew up in a high SES, that does seem to be an influence, as well as comorbid substance abuse. And so for the various outcomes I'm going to talk about, those factors are sometimes very well controlled and sometimes not very well controlled. And that does seem to influence the outcome of the data quite significantly. So I'm sure a lot of you have seen this paper. It got a lot of headlines a few years ago discussing the relationship between cannabis use as an adolescent and this persistent decline in neuropsychological function, where essentially the conclusions of this paper were that if you smoked pot as a teenager, even if you stopped smoking it, as an adult, you still had a decline in IQ because there was these persistent residual effects. This, I mean, I find this amazing. This has gotten an unbelievable amount of publicity. I'm sure if you looked at the altmetric scores on it, it would be quite high. What I find really interesting, though, is now this study has been attempted to be replicated multiple times, and almost every single replication of the study, going more and more into depth on it, has had difficulty finding. They do still see the IQ effect, but they recognize that there are other variables contributing to it. So one study that did longitudinal twin approach to look at this found that the differences in IQ were also present in the non-cannabis using twin, suggesting that there was something about the biology of the individual, less so than about cannabis per se. Another one that was looking at educational outcomes also found differences that related more to substance use. So it's hard, obviously, to isolate cannabis use from a lot of other drugs, such as alcohol and tobacco. This is very important for the brain imaging studies that come up next, but often it's difficult to control for them because the substances are used in a comorbid manner quite frequently. This study claimed that if you controlled for those differences in substance use as well as some other variables, you also no longer saw the effect anymore. And actually the authors of the original paper have now gone back again and published another twin study where they've said that when they look at their twin study, they actually see the same decline, and they think it may have related to some pre-existing differences between the cannabis users and the non-cannabis users. So I think there's a lot of caution to be warranted when we look at retrospective studies in a cross-sectional nature, because we can't control for a lot of the variables that are coming into the mix that could determine differences that exist beforehand. So when we look at brain imaging, we see a very similar problem emerging, I would say. So this was a graph put together by Kent Hutchinson. And what he did was they had done a large-scale study where they did a lot of control for a huge amount of variables that they viewed as potential confounders that would influence the data. And they did neuroimaging indices. And essentially what they were trying to look at was if they could see differences in brain volume, regional brain volume areas, in individuals who used cannabis either as adolescents or only as adults. Their main conclusion, which is the top here, let's see if we can get this to move, was that basically regardless of whether someone started as an adult or as an adolescent, they saw no significant effects on any aspect of brain volume. Now, I'm going to go into their data a bit more in a second, but what they did was then they pulled up every other study that had done this to show the variance in the data across studies. And what you can see here on the well, I guess you're right, on that side over there is data that showed that a brain area had greater volume in marijuana smokers versus non-marijuana smokers. And on the left is showing brain volume areas that were greater in controls than in marijuana smokers. And they have different color codings. The red is the nucleus accumbens, the pink is the hippocampus, the green is the amygdala, and the blue is the cerebellum. So they've reviewed various subcortical brain structures. And essentially what they come up with is that their conclusion is that at the end of the day, a lot of this data is a wash, because if you aggregate it together, you don't see consistency in the regionality of the directional changes, which are likely accounted for by a lot of uncontrolled variables. And so when they did their study, what they made a very strong point about trying to control for was what they viewed as all the moderating variables that would have influenced this data. So they stratified all their cannabis users to non-users. They explicitly took only daily users of cannabis because they wanted to not have the issue of, as Robin addressed, sometimes people just ask, did you smoke or not smoke? So they actually stacked the deck in their favor to find an effect by only using individuals who smoked cannabis on a daily basis. They stratified their users for alcohol because a previous paper that had come out that looked at nucleus accumbens volume, the argument that came out after the fact was that all the data they'd seen was largely accounted for by alcohol consumption. They stratified for alcohol, tobacco, depression and anxiety, and very importantly, in their view, impulsivity and sensation seeking. And I agree with them on this because their argument is if you're looking at individuals who start using cannabis at an extremely young age, they are going to vary on several other traits. It could have to do with the fact that they've had a rough upbringing. It could have to do that they're more impulsive or they're just more sensation seekers. So there's a lot of things that would track with that. So they very carefully staggered or stratified their populations. And this included both controls and cannabis users that had had interactions with the criminal justice system to make sure they weren't biasing in one direction or the other. So they had a very well-controlled study with polarized groups, which in their mind amplified the ability to be able to see if there was going to be an effect. And basically what they found, especially if you look at the p-values, which for the majority of those are in the high 0.9 range, show absolutely no effect. The only effect they saw somewhat of was in the hippocampus, and once they controlled for alcohol consumption, that effect was washed out. So their argument was, at the end of the day, out of the evidence that Ken Hutchinson has generated, and he's done a lot of very interesting stuff down in Colorado now, he argues that if you look at cannabis in isolation, controlling for all the other variables that can influence this, there's no effect on brain growth or brain volume. Now, the one caveat I'm going to put in here about what I view as a potential problem with this is he's not looking at the frontal cortex. And a lot of these structures that he's talking about, I've already mentioned, have developed a lot earlier in life. So it is possible that by not looking at the prefrontal cortex, he has possibly actually missed regional effects that could have emerged in that domain. To that extent, though, Thomas Paus had a large-scale study that came out quite recently that did show that if you looked at polygenic risk score, as Robin had mentioned in his talk, using this kind of index of risk, what you see actually, if you look at a measure of, say, cortical thinning, is that in individuals that, as they progressively load higher on polygenic risk score, you actually do see the emergence of this effect of cannabis use on the cortex, which you don't seem to see at all left of the dotted line in the group that doesn't have any genetic risk to develop schizophrenia. And so I find this quite interesting, actually, because there are a lot of studies that have shown, if you look at schizophrenics who use cannabis versus those that don't, there does seem to be a differential effect to some degree of how cannabis interacts with the brain of someone who has psychosis versus someone who doesn't. This could accelerate damaging problems that would occur that aren't normally seen in the general population. Now, how this would occur, I'm not entirely sure. David Lewis has done some very interesting work looking at differences in CB1 receptor expression in post-mortal brain samples from those who have schizophrenia versus those that don't. So there is the possibility that there is some underlying biological differences, either in the sensitivity or the expression patterns of the cannabinoid receptor in an individual who has schizophrenia that would render their brain more vulnerable to having kind of adverse effects from cannabis use. So I do share Robin's opinion that I don't think that cannabis in the schizophrenic population is a good thing at all, and I think that that's the one area we do tend to see the evidence emerge from in terms of damage there. Okay, so I would say that at this point, from the review of the literature that I've done, as well as talking to people like Ken Hutchinson and the others who've done a lot of these neuroimaging studies, that I'd say there's no convincing scientific evidence that recreation, even with Kent's study, daily use of cannabis in adolescents does have either a residual effect on neuropsychological functioning or regional brain volumes in the average person. Now, obviously that does not exclude extremes in the population on either side, but I think if we're talking about an aggregated data set, I think that it'd be very hard to see a signal there if you actually were able to isolate cannabis as an individual variable. I do think that having genetic risk to develop schizophrenia may moderate that effect. It may make the brain more vulnerable to the effects of cannabis or THC, and you may actually begin to see the emergence of more of a toxic side effect come out in individuals like that. But again, how do we study this in a more meaningful way then? And I think actually the United States has done a very thoughtful approach to this. So there's a large-scale study that they've initiated now about 2 years in called the ABCD study. And what that is Adolescent Brain Cognitive Development Study. And this has about a dozen sites set up around the United States. And what they are doing is they're enrolling 10,000 kids at the age of 9 or 10. Before any of them have started doing any drug use or anything else, well, for the most part, maybe there's a couple. But basically what they've done is they've stratified this across SES, across race, across gender, every variable that they can with a large catchment of 10,000 spread out over different sites, East Coast, West Coast, and in the middle. And basically this is kind of walking through what they do, and they do basic biometrics, looking at blood measures, looking at gene snips, they do brain scans, they do neuropsych testing. They establish a baseline in the kids at about 9 or 10, and then they're going to be tracking them for a 10-year period. And one of the big questions that they actually want to get out of this, because a lot of this was funded through NIDA, whose director is Nora Volk now, is the fact that they're hoping to be able to actually see definitively in a prospective cohort following them if they can, isolating the other variables due to baseline neuropsych functioning and brain volume differences, if they actually will be able to detect differences in those that use cannabis or alcohol or other drugs But the hope is that with the sample size of this large and this much neuroimaging and neuropsych data, you will be able to have big enough ends for users who only go to cannabis or only go to alcohol or only go to cigarettes to isolate the variables, as well as looking at the interacting outcomes that occur from these variables being done in tandem. And they can also control for variables like the impulsivity, sensation seeking, looking at age of onset when the kids actually do start using, and they can track them. So I think this will be a really meaningful data set when it's actually generated. It's a huge expenditure the US government is putting into this, but I think this is kind of what we need at this point to really be able to say we can track someone from A to B and watch them during development and see what they do and see how it modulates their actual trajectory through life. And so I'm very curious to see the outcome of this as it develops. BJ Casey is one of the sites list. I was just chatting with her last week, and it sounds like the ABCD study has almost got it across all its sites, almost all 10,000 kids now. So that's really reassuring. It looks like they have that end to begin with. So I think the next decade will be very exciting for data in this regards to actually see what comes out of this study. Okay. So to summarize what in some ways was a data-light talk, because I'm not exactly sure what to say in this regard. The one thing I will say is I think that there's enough generating evidence that the endocannabinoid system itself evolves during the adolescent period, and that it may contribute to maturation of the frontal cortex and its connections with subcortical structures. So I think from the basic evidence that I've seen from the animal and the limited amount of human studies, I do think there's a signal there. We do need to flesh this out a bit more to understand it better, but I could see that being there. And while I'd say that the preclinical studies do have some significant limitations, like I said earlier on, I do think it's interesting to note that the effects that these studies have reported from adolescent exposure don't seem to be replicated when they do the same treatment regimen in adults. So based on what I was saying before about the biology of the endocannabinoid system in the adolescent brain, I do think that there is the possibility there. Certainly, and I agree with Robin with these escalating levels of THC that are going up and up in cannabis, that is going to become more and more of a problem, I think, because you're going to get more and more activation of this system. And if you have more CB1 receptors in, say, a 13 or 14-year-old in their frontal cortex than you do in someone who's, say, 20, it's more likely that the effect of the drug is going to have on the developing brain will be different than it will on a mature brain. So I do think the potential is there. I just think that, I think possibly one of the reasons the other studies haven't captured it is some of it may be due to the fact that the cannabis that the people were using in those studies was of a lot lower potency. And so it's possible that the signal wasn't there. And I do think this is one of the reasons why, as a country, we need to get a lot of baseline data now as we move into legalization. Not that I think legalization will have major effects on use patterns, but just to understand, because I think if we can quench that high concentration THC from and keep it capped at a certain level, or at least have some regulations get brought into place about, you know, there was a big argument about whether it should be 18 or 25. I think Benedict has created a very sound argument for why it should be the age that was decided upon for the government. And I support that decision, but I think that they're going to have a conversation about that 18 to 25-year-old range. It's going to be to do with potency, because I think if anyone has any concerns, that's where it's going to emerge. And finally, the human studies don't have strong evidence of residual damage, even when use occurs during adolescence. But I think there is a possibility for an underlying interaction with genetics, so that individuals who have a high polygenic risk score for schizophrenia or slight subtle differences in their underlying biology of their system might be at greater risk for effects of cannabis than others. And so I'll just end on, should we be worried? And this is a similar figure to what Benedict had that was data generated from Colorado. And this was looking at legalization happened in 2013 or 14, somewhere in there. And essentially what they found, and this actually was very interesting because at ACMP last year, Nora Volknow was talking about this, and she was genuinely surprised that use patterns among adolescents did not change. There actually was a little bit of an elevation in older populations. I feel that's kind of the baby boomers who are like, we haven't done this for a while, we're going back. So they're getting exposed to something they had when they were younger. But the younger generation already has a relatively high use pattern, and that doesn't really seem to be changing. So I don't think that there is a need for an alarmist like legalization is gonna suddenly cause every teenager to go out and smoke pot. My prediction is that we likely won't see dramatic fluctuations in use patterns. Canada already has, as far as I know, the highest levels of adolescent cannabis use in the world. So there we go. Something to be proud of, I guess. But I think the other point, I just wanted to address the other comment Dominique had made towards Benedict, which is another thing about legalization and access to use. So when she had asked about whether kids would start using other drugs. So my guess is actually, I would say, and this is based on my experience, I grew up outside of Vancouver in the 90s, and what the experience that I noticed was it was quite normal for teenagers to smoke pot. This was a very regular thing in that culture outside of BC. And what actually happened was kids would go, because it was illegal, they would go to a dealer to find cannabis. And once they had a relationship with that dealer then would open the door to them to get access to other drugs. It's kind of what I view as a pipeline effect. No 13, 14-year-olds, at least the average 13, 14-year-old, isn't going out there seeking to go find meth or cocaine or these other hard drugs. A lot of it is incidental exposure because the networks that they move into to access cannabis give them access to other drugs. So my guess is actually I would think that would decline almost, coincident use of other drugs, because if they're able, well, I mean, the question is where are teenagers still going to access it from? My guess is they'd probably still get it from their older brother or something, the same way teenagers access alcohol. But what I think it would hopefully do is if it does effectively kill the black market, those routes that give kids access to harder drugs would be quenched by a legalization process. So I have no idea if this will be an outcome, but that's my guess. I don't know. Well, I guess we'll see. So that's all. Open for questions.
Okay, time for questions. Monica?
I have to ask a biological question, I'm sorry.
Yeah.
I just wanted to, you mentioned that endocannabinoids act as guidance cues very early in development. So this is a real issue of concern of consumption of marijuana during pregnancy. And so do we know anything about this particular aspect and how it might influence or the ability to form the synapses, to guide them, and maybe to prune them? Do we see an accelerated maturation in some circuits and not in others?
Yeah, so Yasmin Hurd and Tibor Harkin did some studies in human fetal tissue from babies that had either not come to term and moms that consumed cannabis, or babies that had died early and moms that had consumed cannabis. They did see some differences in the interneuron arrangement in the hippocampus that was suggestive of a disarrangement that had kind of come out of that. But I mean, as Benedict pointed out, the big thing that's certainly come out is the low birth weight. That definitely is the bigger variable that has been reliable from mothers consuming cannabis. If you look at I mean, Peter Fried in Ottawa had done one of the early large-scale studies where he followed. This was in the 70s. And so moms who consumed cannabis while they were pregnant and then followed the kids, very similar to the way the Dunedin study had done. But the explicit variable being, did mom consume cannabis or not? They did see some subtle effects on executive function. There wasn't anything that was super dramatic. The kids did have a greater likelihood themselves to consume cannabis, although the argument was that could have just been a byproduct of the environment they grew up in. If mom's going to smoke pot when she's pregnant, she's probably going to be pretty liberal about drug use in general. It could have shifted the perceptions in the household, and so the kids could have been more likely to engage in it. There wasn't the profound effects I think that were expected to be seen, and there's been two other kind of larger scale studies that have looked at that, and most of them have kind of come with the same thing. There might be some subtle effects, but they don't seem to be as big as we'd expect, but the low birth weight definitely seems to be the most reliable one.
You mentioned from the data that you summarized that there wasn't a difference in terms of volumes, but you also talked about axonal proliferation and then pruning. Is there a difference in the ways that the pruning happens in adolescence with exposure to cannabis, so that we're not necessarily seeing a volume difference, but maybe structural differences?
I've not seen any data to that extent. I mean, like I said, this has been an area that, given the level of public interest and concern, I'm quite surprised at how little actual science has been done in this regard. I mean, the truth of it was the adolescent brain was understudied up until the late 90s to begin with. So the whole idea of that pruning phenomenon in the scope of neuroscience is actually a relatively new one. And it hasn't been embedded on top of a lot of other things like drug use. So the animal studies have started to kind of looked at it, and the Paralero study that I had cited up there was probably the one that had done the most in animals. They looked, I mean, the end of their argument was actually the effect of THC was very similar to the effect of a CB1 antagonist, actually, which was kind of surprising. And their thought was that because THC is only a partial agonist, but the endogenous ligand 2-AG is a full agonist, that in the presence of THC, it might be blocking the natural actions of 2-AG and acting a bit more like an antagonist, and that's why it's messing up the pruning phase. Now again, their dosing, I think, was quite high. They were using upwards of, I think their final dose that they went to with THC, they would do like a twice a day at 5 mgs and then move to twice a day at 10 mgs and twice a day at 25 mgs, which is really quite a whopping dose of THC. And this is also in a bolus, so you're getting a cloud of THC that's hitting the brain at once as opposed to titered smoking. So I think this is something, I know Ryan with his stuff with the vapor chambers, a big thing they're looking at is dendritic spines in the prefrontal cortex. So they're going to be looking at various adolescent windows. Now again, unless you start doing some of this really high, fancy, in vivo imaging where you monitor the animal through development through like you need to basically get a camera into the brain to track spine changes. You're going to be doing cross-sectional measurements, which is what Ryan will be doing, I think. And I think that will be interesting still because it will be informative, but you can look at dendritic spine profiles in different animals at different stages and see. So it's possible that it's affecting that pruning process.
We have time for another question.
If I can ask you, I'm going home tonight to my adolescence, and this is not what I wanted to hear. What about more subtle effects, other than having the genetic predisposition, the stress diathesis model for schizophrenia? We all know about that. We all see our patients, smoking, and that's the first time they have their first psychotic break, especially the FEP patients, first episode psychosis patients. But what about more subtle effects in terms of what we hear, especially given the higher potency, the less CBD, and things like attention, concentration, executive functioning, school performance, accidents, school dropouts, things that we have on the AMPQ poster, for example. Is there anything you can tell us, that's a bit more subtle, but still, of a concern for our adolescents and for the children of our patients, for example, that we see, for those of us who treat adults?
So I think Gabriella's saying she's going to address some of the subtle effects in her talk. I can't really speak to that because I think subtle effects would be very hard to capture on a broad scale, especially if you try isolating cannabis as a variable, because I mean, especially when you look at the kids that start smoking really young, like if you start smoking pot at 12 or 13, there is probably other variables in your life that are going to make you different than other people. You probably may have had a rougher home life. You're probably going to be trying other substances. It seems very surprising that at such a young age you just start smoking cannabis and not do anything else. You might be exposing yourself to more stressful situations. I think there's a whole host of things that are going to track together. Now, the problem as a scientist, what we do is we isolate variables. And so we want to look at something in isolation. Now, to say that it's the same thing as the comment I made about the schizophrenic brain versus a non-schizophrenic brain, it's possible that cannabis is doing something different in the context of a lot of other adversity going on in someone's life. It might be impacting them in a very different way than, say, it does someone who doesn't have that adversity. But we don't have any information that I'm aware of on anything with this specifically. I mean, there's a lot of anecdotal reports. But I mean, the problem I have with anecdotes as a scientist is for everyone that I talk to that tells me, oh, my son smoked pot and had all these psychiatric outcomes, you can also go be like, well, someone else goes, oh, I smoked pot, now I'm a brilliant lawyer. So it's very hard because they're always countering each other. And you never understand it in the broader context of things. So I do think it's difficult.
Okay,
Sorry, I can't give you more than that.
We are a little bit out of time. We have a little bit out of time.
A little bit out of, are we done?
Yes, thank you.
Okay.